Hello, this is Dhruv Sareen here. I'm from Cedars-Sinai and the executive director of the Biomanufacturing Center. Thank you for the great opportunity to have me present at this webinar along with Miltenyi, where I would be talking about how we can manufacture GMP-compliant induced pluripotent stem cells in closed and open systems. Before I start, I would like to quickly go through my disclosures, which are listed here, and that is our requirement. As an outline to the presentation today, I would like to give an introduction to the Cedars-Sinai Biomanufacturing Center and our cGMP, or Current Good Manufacturing Practices facility. I'll talk a little bit about our capabilities, and then I'll jump straight into our program to manufacture GMP-compliant induced pluripotent stem cells and how we are applying them towards closed cell processing using the CliniMACS Prodigy systems for iPSCs and their derivatives. The Cedars-Sinai Biomanufacturing Center, it's a leading center of excellence in the manufacturing of induced pluripotent stem cells and cell and gene therapies in the growing field of regenerative medicine. We were created with a twofold mission. One is to produce GMP-grade cell therapies to support IND-enabling studies and clinical trials, and to provide patient-specific induced pluripotent stem cells for disease modeling and drug discovery programs. And so we have about 25,000 square foot of clean room GMP, process development, and iPSC research labs all under one roof, right next to and very close to the Cedars-Sinai Medical Center, which is one of the largest hospitals on the West Coast. We have, as part of our cGMP clean room space, we have 9 GMP clean rooms that are certified for ISO 7 and ISO 8. We have 5 standard production rooms with ISO 5 biosafety cabinets, 1 room with cell sorters and the CliniMACS Prodigy, 1 room with the latest large-scale bioreactor systems, and 2 supporting rooms, which are a vial fill room and a media prep room, all to enable manufacturing of cell therapies that would support early phase clinical trials, such as phase 1, phase 2 clinical trials in the space. As we all know, the cell and gene therapy space is growing rapidly, and there's a huge need for CDMO like service providers, whether it's academic or industry-based, really to support all the upcoming clinical trials in this space. We have a lot of supporting rooms, the liquid nitrogen tank, receiving staging, ambient gowning, women lockers, and all of them are equipped with state-of-the-art equipment to enable cell and gene therapy manufacturing. We have a whole host of services that are aligned with supporting the cell and gene therapy manufacturing. So we will produce cell banks. We will do a lot of process development and optimization. We also provide the CMC, chemistry manufacturing controls, IND support for production of these batches and lots. We have an electronic batch manufacturing record system, and we really specialize in manufacturing of iPS cell banks, gene-edited iPS cell banks, and differentiated cells from iPS cells. But in addition to that, we are producing adult stem cells for clinical trials, CAR T-based immunotherapies and NK cell immunotherapies, and harvesting them and supporting automated fill and finish of those cell therapies. We have our compliant QC lab that supports release of many of these cell therapies into the clinic, and we have a robust quality management system that oversees all of these GMP manufacturing and quality control. And then we have process development and iPSC core labs that support many of the activities that are ongoing that will go into the clean rooms for manufacturing. So we are a leading center of iPS cell innovation and excellence. We have a research side where we're supporting a lot of work on creation of iPS disease models, whether it be for neurodegenerative, cancer, pancreatic, vascular diseases. We support manufacturing of cells for tissue engineering, 3D bioprinting, organ chips. But really, we are at the cutting edge of creating these cells for treatment of diseases for regenerative medicine and supporting IND-enabling clinical trials. And as we all know, cell therapies encompass a wide range of products, whether it's autologous product, single lot, an allogeneic product, large scale, and bank based. They both have common concerns that are related to the mechanism of action of cell therapies, the material qualification, the critical quality attributes, the facility, product shipping handling, and the challenges and timelines that are associated with that. But each of them have their own specific concerns, right? On the autologous side, we have issues related with product variability, batch to batch from production of that, limited material and time for testing, a short shelf life, and the logistics and scale out issues. On the allogeneic side, we have to think about donor eligibility, qualification of those cell banks, stability of the cell banks and intermediates, the scale up, genetic integrity of those cell banks. And so iPS cells fit actually largely on the allogeneic product and large scale bank side of the spectrum, but there are programs that are using autologous iPS cells for production of gene edited cell therapies. And so they really encompass a wide array of spectrum. And over time, we do see a lot of iPS platforms moving towards an autologous platform as well. So the power of induced pluripotent stem cells is that they can really generate any cell type in the human body. We can make blood cells, muscle cells, all the neural cell types, fibroblasts, pancreatic cells, epithelial, and all of them are across the world are being used for multiple clinical programs. And our goal is to really enable that and be a piece of the puzzle. We have developed our own method for episomal-based reprogramming of unexpanded peripheral blood mononuclear-derived iPS cells. And we see really high success rates of reprogramming using this method. And we have developed a xeno-free method without requiring mouse embryonic feeders and all GMP-grade reagents and animal origin-free media to expand and create these iPSC lines. But what is really unique about the iPSC lines that we make using our method is they have a really high enhanced rate of cytogenetic stability with these unexpanded PBMC-derived iPS cells. What you see on this slide is what we call an ideogram, a chromosome ideogram, that shows all the chromosomes from chromosome 1 to 22 and the sex chromosomes X and Y. Just traditionally, it's known from human embryonic stem cells and cells that are derived from expanded source tissues, such as either expanded blood cells or fibroblast cells, and cells that we make, iPS cells that we make from unexpanded PBMC-derived iPS cells, that we have almost a four-fold lower rate of karyotypic genetic abnormalities that are seen over the timeline of the genesis of those iPS cells across multiple passages. And if we exclude sex chromosome abnormalities, it's actually a very low rate of abnormality. And we've now karyotyped close to 2,000, over 2,000 G-band karyotypes across expanded and unexpanded source tissue-derived cells. So now with this method, we are taking our internal programs as well as our clients' programs to derive iPS lines in GMP-compliant manner. In this slide, we have published a review and cell and gene therapy insights where we show sort of a pipeline of manufacturing these iPS cells. We take a simple blood draw from the donors, obviously consented appropriately and performing all the required donor eligibility criteria as per 21 CFR Part 1271. And then we go ahead and create and isolate those PBMCs. We cryopreserve the PBMCs, and then we can reliably reprogramming using our episomal plasmid-based method. We can expand those cells either as adherent cell cultures, or if we need to scale up in a high volume in these spinner-style bioreactors. And now we're working on process development to adapt that to larger 20-liter and 50-liter scales over time. So we, using this platform, we procured funding from the Department of Defense. The solution was to create a GMP-compliant iPSC haplobank. What is an iPSC haplobank? An iPSC haplobank is made of human leukocyte antigen haplotype homozygous donors that can provide HLA-matched material to significant numbers of patients. Cells derived from these donors may still require the use of immunosuppressive agents after transplantation, but data from clinical and organ and cell transplantation shows a clear benefit of HLA matching to minimize rejection. HLA matching between donor and recipient limits T cell activation, and using HLA haplotype homozygous donors will reduce direct T cell activation against iPSC-derived cells or tissues. However, as other elements of the immune response, such as NK cells and minor histocompatibility antigen reactive T cells, may elicit some rejection responses, and it is likely that still some degree of immunosuppression will be necessary when using tissues or cells derived from these HLA homozygous iPSC lines. So for this project, we considered HLA-A and HLA-B loci because HLA-A and HLA-B are constitutively expressed at higher level in most cell types. So we will require a much greater number of homozygous donor iPSC line if we want to match at all the HLA loci. However, this was just a start to establish a GMP-compliant iPSC haplobank with this project. And we really expect that the next stage of this project, as the global iPSC market is expected to reach almost $4 billion by 2024 with a compounded annual growth rate of 10%. From this GMP-grade iPSC haplobank, we wanted to prove that we can create a derived cell product that could be used in military applications. So military combat injuries often involve damage to organ and tissue vasculature, and the rate of vascular injury in modern combat is about 5 times the previous wars. So endothelial cells was a good template, and they can be really used in regenerative medicine for ischemic and sepsis conditions, as well as for orthopedic applications. And on engraftment, we expect endothelial cells to induce significant revascularization and contribute to repair of the damaged tissues and bones. As a result, the vascular cell and graft market is poised to reach over $1 billion by the end of 2025 at a compounded annual growth rate of about 5 to 6%. So using iPS-derived endothelial cells is of significant advantage because there's a major limitation of using other somatic cell types, such as cord blood or peripheral blood, to isolate endothelial cells. And the feasibility of such an approach lies really in this insufficiency of the cellular numbers required for such a therapy. Extrapolating from animal studies, an estimated 12 liters of blood would be required to generate endothelial cells for effective treatment of ischemia in average adult patients. Such cells from peripheral blood or cord blood do not really scale rapidly and lose the ability to replicate in sufficient numbers. So therefore, iPSC-derived endothelial cells really provides a readily available off-the-shelf solution. As a result, we have developed from iPS cells to vascular endothelial cells a GMP-compliant protocol to generate such vascular endothelial cells. It is quite a simple protocol, and we have really optimized it over the last 2 to 3 years in order to take out all animal components and really create a xeno-free protocol that is an adherent-based protocol and provides high efficiency of endothelial cell differentiation. And we can make, for example, as shown in the slide, CD144 and CD31, which are canonical markers of vascular endothelial cells, really at a high degree of efficiency from multiple iPSC lines using this GMP-compliant protocol. So one of our goals was in this method is how can we create these cells in a closed cell processing system like the CliniMACS Prodigy. It's important for any iPSC-derived cell therapy development to take it from a lab scale where it's an open system to a scale where we can try to get it into a closed system. The importance of that is, one of the greatest benefits of using a closed loop system is the reduction of risk of contamination through exposure. Closed systems provide a physical barrier to prevent contaminants from affecting the product, while open systems provide an opportunity for contamination just simply through the process of being open to the room environment and physically being handled by operators. So avoiding contamination is important not only for safety reasons, but also because contamination can lead to costs associated with product loss and cleaning. The other benefit of an automated closed system cell therapy manufacturing is it requires experienced professionals to conduct these steps in the process. So closed systems, on the other hand, don't rely as much on operator handling and thus are less prone to human error. In addition, the full automation of the process eliminates all these extra steps and making the processing time much faster. The other big advantage is automated closed systems. There's a much greater cost effectiveness. There's lower overhead costs, such as reducing the environmental monitoring and air handling requirements, energy savings by reducing these costs, removing or reducing gowning requirements, also reduce the cost of both gown materials and provide time savings for gowning and the degowning process. So there's a lot of overhead costs in cell therapy manufacturing, and we're all trying to get those defined labor costs out of cell therapy manufacturing, and these closed systems provide a great advantage. So our goal was to see if we can take iPSC culture and iPSC-derived endothelial cell differentiated culture and adapt it to a closed system process. So the CliniMACS Prodigy is quite suited for that, right? It's well known, and there's commercially approved products, especially in the immune cellular and immunotherapy CAR T manufacturing space, that require the CliniMACS Prodigy and have already been used for many of those applications. We wanted to see if we can use customized applications that are tailor-made for automating GMP-compliant solutions for final cell product or cell banks of iPS cells or iPSC derivatives. And so we worked with CliniMACS Prodigy and Miltenyi to take their Adherent cell culture platform to see how we can get high yield production for our GMP compliant protocol to manufacture iPSC-derived endothelial cells. And we did this by using the Corning cell stacks, first by using the single layer stacks, trying to get the process development piece in place. And so the CliniMACS Prodigy has a tubing set that is specifically designed for Adherent cell culture platform, which we can use either in these chambers that you see on the side or the cell stacks that are designed for Adherent cell culture manufacturing. One layer of a cell stack provides about 636 centimeters square of space, whereas the vessel provides about 100 centimeters square of space for cell growth. We worked on adapting the CliniMACS Prodigy Adherent cell culture system to develop this closed system for iPSC-derived endothelial cell production. And here are different steps in the Adherent cell culture platform overview that requires either pre-processing, coating of the flasks, inoculation of the cells, harvesting, dissociation, and we had to utilize a combination of all of these to get to sort of an optimal platform for iPSC culture and differentiation to endothelial cells. And so here is a credit to our scientist, Arvind, who's worked with Miltenyi to adapt this platform with the tubing set 730 for the CliniMACS Prodigy Adherent cell culture platform. And here she's working on loading and adapting the system towards our process. We require the external cell vessels, which is in this case is the Corning cell stack, to be sterile welded to all the tubing sets, and that requires to all the bags that contain the coating, dissociation, neutralization buffers, they have to be sterile welded. And so we use all of these platforms to sterile weld that connects to the external cell vessel via a 1 millimeter tube extension, and all unnecessary attachments that can also be sealed sterilely. And this is us initiating the CliniMACS Prodigy system for endothelial cell differentiation. And here in the image on the right, you can see all the tubing sets and the media loaded and the Adherent cell culture platform connected to perform iPSC culture and then subsequently differentiation into this platform. And then ultimately, the idea is we can increase the scale and yield to 5-layer cell stacks, and then eventually we can even connect 3 of those 5-layer cell stacks in series or in parallel to perform high yield IEC differentiation. Here's some images of the cells that are growing in this platform, actually grown on the CliniMACS Prodigy in the single-layer cell stack using this metric. So we seed the iPSC cells as small colonies at a relatively optimal cell density that we've defined. And here is the colonies that grow in these cultures. We grow them for 3 days as iPS cells before initiating differentiation, and that is required as per our protocol. And once we initiate the differentiation, so that's from day -3 now to day 2, so in total 5 days of differentiation in the cell culture platform, these cells start looking, these colonies start differentiating into mesodermal and then vascular cell progenitors. So we experience close to an 80 to 100-fold expansion during this period, going from day -3 to day 4, which is total 7 days of expansion to the vascular cell progenitors. Here they are at day 4. This is the point when the cells are harvested and then reseeded onto a new flask with a new substrates that defined for specifically for endothelial cell differentiation. And ultimately, over time, this is what the cells look like. Here they are in vascular endothelial cell progenitors, and they go through a 5 to 10-fold expansion during the stage. And here they are more images of them getting more confluent in this platform at day 11. So we're very happy to report that we've successfully been able to adapt this platform at a slightly medium scale in a single stack using the CliniMACS Prodigy and Adherent platform. And we look forward to continuing to work to increase the yield and the scale of endothelial cell differentiation from iPS cells, and even for iPSC culture using this platform. And then ultimately move it to the clean rooms where we can have a completely automated and closed system process. In summary, the Cedars-Sinai Biomanufacturing Center was created to address a bottleneck in the capacity of cGMP cell manufacturing. We produce research-grade and GMP-grade patient-specific induced pluripotent stem cells and derivatives, as well as GMP-grade cell therapies for IND-enabling clinical trials. We have developed special expertise and technologies in reprogramming unexpanded peripheral blood mononuclear cell-derived iPS cells that would provide starting material with excellent genetic stability and would be a scalable solution for cell therapies across the world. We are developing now a process to reprogram and create an iPSC haplobank, which would provide a more feasible off-the-shelf solution that can be scaled out, and showing that as a proof of concept, we can make GMP-compliant iPSC-derived vascular endothelial cells for vascular injuries such as ischemia or sepsis. Using the CliniMACS Prodigy Adherent Cell Culture System platform, we are creating iPSC-derived vascular endothelial cells in a closed system, and this system has its own unique advantages. With that, I would like to close by saying none of this would be possible with a talented and dedicated team at the Biomanufacturing Center. Some of them are pictured here. And the funding from our institutional funders, the Cedars-Sinai Board of Governors, as well as the Department of Defense and the Medical Technology Enterprise Consortium that has funded some aspects of the program I presented today. With that, here's a link to our biomanufacturing website. And thank you, and I'll take questions later. I'll pass it on to my colleague at Miltenyi, Dr. Chao Sheng, to present his portion of the talk.
Many thanks, Dr. Sareen, for the great talk. And I think really the works you just presented on GMP-compliant human iPSC manufacturing processes at the Cedars-Sinai Biomanufacturing Center set really great examples in the field. And I think we can all learn a lot from your work. So my name is Chao Sheng, and I'm the Global Program Manager for the Regenerative Medicine Manufacturing Portfolio at Miltenyi Biotec. So now I will also just very quickly introduce the established workflows at Miltenyi Biotech for automated closed and GMP-compliant manufacturing of human pluripotent stem cells. So then I can also give a bit more technical details about this CliniMACS Prodigy adherent cell culture process. So speaking of manufacturing of PSCs and their derivatives, so we all know that these are very challenging, very complex endeavors. And most of these current manufacturing processes, they involve multiple expensive devices, very skilled operators, and also high-class clean rooms. And many separated open handling steps are also introduced into these workflows. At the same time, we also know that standard in-process control and quality control analysis are required throughout the entire manufacturing processes. And in addition, so in certain applications, the desired cell populations also needs to be further isolated in a sterile, gentle, and GMP-compliant environment. So all these factors together could really limit the manufacturing skill, compromise the reproducibility, at the same time also drive up the manufacturing cost. So how to make these very complex cell manufacturing processes more standardized and more scalable, at the same time also more affordable for the end users? So these are some solutions from Miltenyi Biotec. Yep. First, with the CliniMACS Prodigy platform. So these separated open handling steps, these multiple devices, skilled operators, and high-class clean rooms, they can be integrated into one automated and closed system. And the users can also build customized PSC expansion and differentiation workflows on the CliniMACS Prodigy. And then with the MaxQuant analyzer, so standardized iPSC QC analysis can also be realized by automated flow cytometry analysis. And finally, the MaxQuant title cell sorter can be applied to isolate the desired cell populations with a very gentle, fast, and also GMP compliant cell sorting technology. So today, we will focus on the CliniMACS Prodigy platform. So together with these disposable tubing sets, the CliniMACS Prodigy allows automated cell processing from starting material to the final product. And upon your request, it can automate all these different cell handling steps, including sample preparation, density gradient centrifugation, cell washing, medium change, etc., until the final product formulation. And based on this CliniMACS Prodigy platform, we have already developed a broad range of software applications for various cell types and workflows. For example, the CAR T cells. Well, for the adherent cell types, like pluripotent stem cells and their derivatives, then we have developed this adherent cell culture process. So using the CliniMACS Prodigy adherent cell culture process, then the cell cultivation can take place in two different units, the centrical unit and the external culture vessels. Yeah. First, there is a CliniMACS Prodigy chamber lies inside the centrical unit. It supports automated adherent and non-adherent cell cultures. The centrical unit supports controls of temperature, the air, and CO2 concentrations. There is an integrated microscope under the chamber for visual examinations during cell manufacturing. And there are also multiple input ports for using different medium and cytokines. And this centrical unit offers 100 square centimeter surface space. Well, if larger surface space is required, then these external culture vessels, such as the multi-layer cell stacks, can also be easily connected to the tubing sets 730 via stereo welding, as Dr. Sareen just mentioned. So the users can connect actually maximally 3 of these 5-layer cell stacks at the same time. And then this will offer roughly 10,000 square centimeter surface space, so already fairly large. So now let's also have a look of this adherent cell culture software. So when we designed it, our goal is to make sure that it's flexible enough to culture different cell types. So that's why we modularized the individual cell handling steps. And now on the main menu of this CliniMACS Prodigy adherent cell culture process, you can find all these different modules for density gradient centrifugation, for coating, inoculation, cell culture, medium change, and harvest. And the users can freely select and combine these different modules in different orders. And as a result, very complex workflows, such as expansion and the differentiation of human pluripotent stem cells, can be realized on this CliniMACS Prodigy adherent cell culture process. And within each module, so the users can also set up the cell culture parameters according to their established SOPs. For example, in the coating module, the users can set up the coating temperature, the coating time, the volume of the coating reagents according to their established protocols or SOPs. So we have already established several workflows on the CliniMACS Prodigy adherent cell culture process. Yeah. So this includes the GMP-compliant expansion of human MSCs and PSCs, as well as directed differentiation of human PSCs into mid-brain dopaminergic progenitors and cardiomyocytes. So now I will just very quickly show you some performance data on the GMP-compliant PSC expansion workflow. So for this workflow, we have first developed the iPS-Brew GMP Medium, so which is manufactured and tested under the quality management system ISO 13485, and is also designed following the recommendations of USP 1043 on ancillary materials. So it is currently available in bottles, but soon we will also offer this medium in bag format, so they will be very compatible with the closed CliniMACS Prodigy system. And in our experiment, the results shows that this medium enables very stable and very efficient human PSC expansion as either small clusters or as single cells. So it also supports very fast culture reinitiation after cryopreservation. And it is actually based on the same formulation as our research-grade StemX iPS-Brew xeno-free medium. That's why this can support seamless transitions from research-grade to GMP-compliant processes. Then, by applying this iPS-Brew GMP medium on the closed and automated CliniMACS Prodigy adherent cell culture process, then the users can choose and combine these different modules to automate the complex handling steps. For example, coating of the culture vessels, PSC inoculation, medium change, harvest, etc. And we have also performed side-by-side comparisons between manual and the CliniMACS Prodigy-based PSC expansion using our flow cytometry iPSCs technologies. And looking at the expression levels of these pluripotency markers and the trilineage differentiation capacities, so the CliniMACS Prodigy and the manual control groups, they are very comparable. And by using the centric unit and the different sizes of the external culture vessels, then the users can also easily reach different PSC expansion scales with much less open handling steps. Yeah, and we all know that establishing these GMP-compliant PSC banks is a major step in many clinical applications. And for this purpose, the closed and automated CliniMACS Prodigy adherent cell culture process, together with the iPS-Brew GMP medium and our flow cytometry iPSCs technologies, so they could be very valuable asset for our customers. So here we have another example. So recently, Dr. Raphael Kroll from the Sarah Foundation in Japan has also successfully transferred their manual clinical grade human iPS expansion workflow into the CliniMACS Prodigy adherent cell culture process. So he has presented his work at the ISCR 2021 Tokyo Symposium and also at a joint webinar with us last year. So these presentations, the webinar and the posters, they were available on our website. So you can please feel free to have a look. So actually, that's already everything from my side today. So this is the disclaimer of my presentation. So in the end, thanks a lot for your attention, and also many thanks to Dr. Sareen for the very insightful presentation.